Hello everyone, this is Dr. Nasser, and in this clip, uh, I would like to tell you about thalassemias. Specifically, in this smear, we have beta thalassemia minor. So, thalassemias in general are a diverse group of genetic disorders that, uh, in which the production of hemoglobin protein is going to be either inhibited or reduced. And as you are aware of, there are different hemoglobin genes because the hemoglobin molecule is going to be a tetramer, consists of 4 different proteins. Now we have different type of hemoglobins: adult hemoglobin, fetal hemoglobin, hemoglobin A2, and other hemoglobins that we have during the fetal and embryonic age. However, the important fact that you need to realize about thalassemias are diseases that there is going to be an imbalance in the production of the hemoglobin genes. So if there is an imbalance, there is always going to be the chance for the hemoglobin molecules to bind to each other the way that they are not supposed to. So you would be able to see that 4 alpha globin chains are coming together and making a hemoglobin molecule, which is going to be abnormal, and that hemoglobin molecule will precipitate in cell and subject the cell to early destruction as it goes through the spleen or encounters macrophages. In this field, what we are seeing is a lymphocyte, slightly large lymphocyte. It is not a resting lymphocyte, but I would say that the size of this lymphocyte is going to assist us to detect a general characteristic of all these cells. These are all microcytic cells. And if you notice, you will see that most of the cells are the same size. Because remember, this is a hereditary problem. Uniformly are going to have smaller size than the nucleus of a resting lymphocyte, but this nucleus is not much larger than the resting lymphocyte. So there is microcytosis that is present on this smear. You always see in hemoglobinopathies as well as in thalassemias target cells. So target cells are also present in this smear. You may be able to see other abnormal red blood cell morphologies. There is a spherocyte in here. There is a schistocyte in here. And as we move through the field, we do see that, yes, target cells are consistently present across all fields. Here we have an elliptocyte, another spherocyte, another spherocyte. And I would say that in this case, the patient is going to demonstrate minimal symptoms. And those symptoms usually are going to be characterized by about hemoglobin around the range of 12 to 14 or 10 to 12, depending on whether you're a male or a female. And hematocrit is going to be lower on the CBC. The MCV is going to run about 75 femtoliter. You may be able to see hypochromia. Of course, in this field, we don't have any hypochromia, but as I go to other fields, I do observe that these cells display significant amount of hypochromia. So cells in thalassemia are going to be small in size, microcytosis, and they are going to be also hypochromic. There are Possibilities that you have other abnormal morphology of the red blood cells. There's a dacryocyte in this corner, and basically you have to review several fields, and when you see a morphology that is persistent in several fields, then you would be able to include that in your report. For this report, I will indicate the hypochromic microcytic cells. I would indicate the presence of target cells. I would perhaps indicate the presence of abnormal morphologies such as spherocytes, schistocytes, as well as elliptocytes. But in general, you may see increased polychromasia, but that increase is not going to be very much. Either the reticulocyte count is going to be normal or slightly elevated.